The Epstein-Barr virus is a typical herpes virus that has a unique ability to establish latency and long-lasting infections. EBV is the culprit of many common diseases, such as infectious mononucleosis, Burkitt's lymphoma, and nasopharyngeal cancers. Viral persistence is established through the cycle of infection, along with exploitation of the body's B cell functions. EBV is transmitted in saliva. Early infections are almost always asymptomatic. The primary lytic infection is initiated in the oropharyngeal epithelium. While the early steps are not well characterized because of a 4 to 6 week incubation and latency period, it is still unknown whether the virus originates in the B lymphocytes of the epithelium or the epithelial cells themselves. During the incubation, the EBV viral particles effectively bind to CD21 receptors found on B cells. Once this happens, the virus goes latent. During latency, B cells express different genes and proteins. The proteins will then drive the proliferation for the latency-infected B cells. EBV viral particles pass along their genomes to daughter cells as the B cells divide. EBV has potential to shed into the tonsillar or pharyngeal epithelium and then reside in one's saliva, which then could be transmitted between people. The process of B cell division could also trigger an immune response, potentially preventing the individual from experiencing any symptoms. On the other hand, the Epstein-Barr virus can cause an immune response in by which an EBV-specific antigen can capture the viral particle and process it within the B cell. The B cells will then enter the bloodstream and make its way to the lymphatic system, where it causes another immune response, either within the lymph node itself, the liver, or the spleen. Personally, when I had EBV, infectious mononucleosis, my spleen, liver, and lymph nodes were all enlarged on scans. Once inside the lymph nodes, B cells promote CD8 T cell binding. CD8 T cells are also known as cytotoxic T cells, which are released into the bloodstream and help prevent reactivation of EBV. B cells will also bind to CD4 cells, which in turn activate each other. After B cell activation, B cells will convert into plasma cells, which will in turn release EBV-specific antibodies. There is the potential that EBV viral particles are released into the bloodstream after reactivation. The presence of EBV particles will become recognized by macrophages within the epithelium, which will hence release cytokines and initiate viral infection, producing symptoms. As mentioned prior, EBV has the potential to affect other organs within the body by traveling within the bloodstream/lymph system. Abnormal side effects like elevated liver enzymes and bilirubin levels should be expected.